What makes you, you? What gives humans the ability to write poetry, build rockets, and crack jokes? The answer lies in this remarkable 3-pound universe between our ears. It's roughly the size of a large cauliflower. This remarkable organ powers everything that makes us human. Our brain makes up just 2% of our body weight while consuming a whopping 20% of our body's energy. Imagine if your cell phone used 20% of your home's electricity. That's the kind of energy demand we're talking about. Let's put this in perspective with a journey through the animal kingdom. Picture yourself in the zoo, walking past different exhibits. The elephant, massive and intelligent, has a brain 4 times heavier than ours, about 12 pounds. Next to it, a bottlenose dolphin with a brain slightly larger than ours at 3.5 pounds. Then we see our closest relative, the chimpanzee, with a brain weighing less than a pound. Moving to the domestic section, we find our house cats with brains the size of a walnut, about 1 ounce. And finally, a mouse with a brain weighing less than a paperclip. Inside those brains, the numbers are staggering. Our human brain houses an incredible 86 billion neurons. That's more than 10 times the numbers of people on Earth. A chimpanzee has about 28 billion, while a mouse brain contains just 71 million. To put that in perspective, we have about 1,000 times more neurons than a mouse. You might think this explains everything about our superior intelligence. But hold that thought. Let me introduce you to Raven. With a brain the size of a walnut, ravens can solve complex puzzles, use tools, plan for the future, and even remember human faces. The raven's brain weighs just 10 grams. Even more remarkable is the humble honeybee. With a brain smaller than a grain of rice, just 1 cubic millimeter, bees can navigate complex routes, communicate through intricate dances, remember flower locations, and make collective decisions. They're basically running complex GPS systems with a brain the size of a pinhead. And then there is the octopus, nature's 8-armed genius. Instead of a centralized brain like ours, they have 500 million neurons distributed throughout their arms. It's like having a brain in each arm working together to solve problems. So if size is not everything, what is it? The answer lies in organization. Think of it like cities. A large city is not automatically more efficient or productive than a smaller one. What matters is how well it's organized, its infrastructure, its networks, its systems. Our brains are like incredible, well-organized cities with dense networks of connections and specialized regions working together. This organization comes at a cost though. Remember that 20% energy consumption I just mentioned? Ravens pack more computing power per gram than we do. Their brains are like highly efficient, compact computers, while ours are more like powerful supercomputers that need massive amounts of energy. Imagine walking into 2 different restaurants, one Italian and one Japanese. Both kitchens might stock remarkably similar ingredients: flour, water, salt, and other seasonings. However, what comes out of those kitchens can be dramatically different. This is exactly what we're discovering about the human brain compared to other species. When scientists first started mapping brain cells in detail, they were actually surprised by how similar the basic cell types were across different species. Just like those restaurant kitchens, it's not so much about having unique ingredients, it's about how you use them. Humans and mouse brains have essentially the same types of cells. It's the proportions that make all the difference. Think about how both a light, airy croissant and a dense, chewy bagel can come from largely the same ingredients. In the human brain, particularly in areas like the auditory cortex, People have 5 times more support cells compared to neurons than mice do. These support cells, like astrocytes, oligodendrocytes, and microglia, are like the kitchen staff that make a fine dining restaurant run smoothly. Astrocytes are like skilled prep cooks. They maintain the environment around neurons, provide nutrients, and help form connections. Oligodendrocytes are similar to the insulation around electric wires in your kitchen appliances. They speed up signal transmission and protect the connections. Microglia are the cleanup crew, removing damaged cells and pruning unnecessary connections, much like a chef trimming away excess fat or removing wilted leaves. When it comes to brain development, humans are remarkably slow compared to other species. This may seem like a disadvantage at first. After all, a newborn deer can walk within hours of birth, while human babies take about a year to achieve the same feat. But this slowness is actually one of our brain's greatest strengths. Think about how a fast-growing bamboo shoot reaches its full height quickly but remains relatively simple in structure. By contrast, an oak tree grows slowly over decades, developing intricate branch patterns, deep roots, and remarkable resilience to environmental challenges. Our brain follows a similar principle. Human neural progenitors, the cells that eventually become neurons, spend much more time in the developmental stage that scientists call limbo compared to other species. During this extended period, those cells have more potential to become different types of neurons, unlike in rodents where each progenitor typically develops into just one type of neuron. This human brain developmental journey is remarkably long and complex. While a baby is born with almost all the neurons they'll ever have, about 86 billion of them, the brain is far from finished at birth. However, those neurons are like extensive network of roads that have not yet been connected by bridges and highways. What follows is one of nature's most remarkable construction projects. During the first few years of life, those neurons begin forming connections between neurons called synapses at an extraordinary rate. By age 3, the brain has formed about 1,000 trillion synapses, which is twice as many as will be present in adulthood. This massive proliferation of connections creates a dense network of neural pathways that far exceeds what we see in adult brains. Think of it like a sculptor starting with more marbles than needed, knowing that the final masterpiece will emerge through careful refinement. This surplus of connections then undergoes a process called synaptic pruning. Like a gardener carefully trimming away excess branches to help a tree grow stronger, the brain eliminates unused or less efficient connections while strengthening those are frequently used. This pruning process is not random. It's guided by experience and environmental input, which ensures that the neural circuits that remain are optimized for the child's specific environment and needs. Different brain regions mature at different rates. Sensory processing areas develop early, while the prefrontal cortex, responsible for complex decision-making and emotional regulation, is not fully mature until around age 25. This early development of abundant neural connections creates a critical window for learning. Research has shown that children who are exposed to multiple languages before age 3 can acquire them with remarkable ease, which develop native-like proficiency in each. This is because their brains are especially receptive to language input during this period, with neural circuits readily adapting to process different sound systems and grammatical structures. The same principle applies to other forms of learning. Diverse experiences during those early years, whether through music, movement, or social interaction, help shape robust neural networks that can persist throughout life. For example, children who experience rich sensory environments and different learning experiences before age 3 tend to develop stronger cognitive abilities and greater adaptability in learning new skills later in life. It's as if the brain during these early years is like a highly sophisticated recording device, eagerly absorbing and incorporating every experience into its developing architecture. The environmental influence on our brain development is particularly important because our brains take so long to mature. They can be shaped by our experiences, relationships, and learning in ways that faster developing brains cannot. This is why human children can adapt so readily to different cultures, languages, and environments. It's like our brains comes with a longer programming period that allows them to be optimally configured for whatever environment they find themselves in. This extended plasticity is especially evident in language development. Children can easily become fluent in multiple languages if exposed to them before puberty. This becomes progressively more difficult as the brain continues to mature. I started learning English as a second language when I was 12 years old. At that point, my brain had already gone through extensive synaptic pruning, particularly in areas involved in language processing. It became challenging to have native-like pronunciation because it was difficult to pick up subtleties in the new language, especially phonetic aspects. This is part of why accents and certain grammatical structures can be more difficult to acquire fully after the critical period, which typically spans from birth to about age 7 and started to decline noticeably by age 12. The critical period of basic visual processing circuits generally takes place in the first few years of life, often peaking between birth and age 2. During this time, the brain is highly responsive to visual stimuli, which allows for the development of essential structures needed for depth perception, color differentiation, and visual acuity. If a child's visual input is disrupted, for example, due to cataracts, nearsightedness, and farsightedness, this can lead to long-term deficits in visual processing that are difficult to correct after this early window. For the emotion regulation systems, the critical period extends further into early and middle childhood, approximately from birth through age 5 to 7, though it remains somewhat plastic into adolescence. During this time, children learn to process, express, and regulate emotions through interactions with caregivers and their environment. This early period is particularly important for forming secure attachment bonds and learning adaptive emotion regulation strategies. Disruptions during this time can increase a vulnerability to mood disorders and difficulties with self-regulation of emotion later in life. The brain's 2-decade development comes with its own set of vulnerabilities. The elaborate neural networks that enable our advanced cognitive abilities also create more opportunities for things to go wrong. This is why humans experience unique conditions like schizophrenia, bipolar disorder, and other forms of dementia that are rarely, if ever, seen in other species. The prolonged development of our prefrontal cortex while enabling sophisticated cognitive abilities also create an extended window of vulnerabilities to psychiatric disorders. For example, schizophrenia typically emerges in late adolescence or early adulthood, precisely when the prefrontal cortex is undergoing its final stages of maturation. The complexity of our neural networks also makes them more susceptible to cascade failures. Small disruptions in key developmental processes can have far-reaching effects across multiple brain systems. Subtle changes in synaptic pruning during adolescence had been linked to the development of schizophrenia. Similarly, the sophisticated circuits that enable our advanced social cognition and emotional processing makes us particularly vulnerable to mood disorders. Conditions like major depression are notably more prevalent and complex in humans than in any other species. This vulnerability is further compounded by our uniquely long lifespan. Humans live well beyond their productive years, putting unprecedented demands on brain maintenance systems. Research comparing human and primate brain aging has shown that humans experience more severe age-related neural deterioration, particularly in regions supporting high cognitive functions. This may explain why advanced age is the greatest risk factor for neurodegenerative conditions like Alzheimer's disease, which appears to be uniquely human in its full manifestation. The prefrontal cortex does not reach its full structural and functional maturity until around age 25. However, this same brain region begins showing subtle signs of decline relatively early in adulthood. Studies use brain imaging have detected measurable volume loss in prefrontal cortex starting in our mid-40s, decades before other brain regions showing significant age-related changes. By age 60, the prefrontal cortex typically shows about 10% to 15% reduction in volume compared to its peak size in adulthood. This shrinkage is not uniform. It particularly influences the dorsolateral prefrontal cortex, a subregion crucial for working memory and complex decision-making. The abilities that took longest to develop in your youth, like complex decision-making and emotion regulation, are often the first to show subtle declines. But don't worry, our brains are remarkably adaptable. The skills you've thoroughly learned and practiced over your life typically remain strong and stable. Our brains develop compensatory mechanisms. Think of it as a business restructuring. When the CEO, which is prefrontal cortex, starts becoming less efficient, other departments step up to help. Older adults often recruit additional brain regions to maintain performance on different tasks. The backup systems are ready to kick in when needed. How to make sure we have robust backup systems? Stay mentally active, engage in learning new things, maintain social connections, and stay physically active. Those activities help build what scientists call cognitive reserve. When we engage in challenging mental activities, especially learning new skills, our brains not just strengthen one pathway, it creates multiple routes to accomplish the same task. Think of it like learning to drive to work. The more routes you know, the less likely you are to be stopped by a single road closure. Studies have shown that bilingual individuals, for example, often show delayed onset of dementia by 4 to 5 years compared to monolinguals, precisely because they've developed multiple neural networks for language processing. Learning a new language strengthens connections between memory and language areas. Playing a musical instrument builds bridges between motor, sensory, and cognitive regions. Social interactions enhance networks associated with emotional processing and decision-making. Physical exercise increases the blood flow and promotes the formation of new neural connections. Each of those activities contribute to what researchers call neural flexibility, the brain's ability to recruit different regions to maintain performance when primary pathways begin to decline. So what makes you, you? It's a combination of biology, experience, and the choices you make to keep your mind engaged, enriched, and active. Every time you push yourself to learn something new, solve a problem, or listen to people who disagree with you, you are helping your brain stay strong, flexible, and uniquely you.